So today we are going to be talking about neurofeedback and some EEG and fMRI. You have had a couple of lectures already on EEG and fMRI. So first of all, I'm not going to get into details of that, though if anybody had questions from whatever was talked in the last two lectures, I'm happy to answer and wrap up on that. And and second, most of the things that I will show not necessarily will have EEG and fMRI simultaneously. Actually, most of it would not, though we are going to discuss a little bit on how to combine the things. But mainly we are going to focus on neurofeedback. So probably all of you are familiar with what feedback is and what biofeedback principle is. Here is a schematic. Actually, Pete here was not running an experiment, but rather submitting a paper. But we modify this. In a regular experiment, you will have a subject that is trying to correct some physiological parameter, right? And these will be done based on some information that we are giving to him or her. So for that, we need to measure a physiological parameter. The data needs to be sent to a computer, do some analysis, extract some information, and that's what we display back to the subject. Would be kind of the similar idea if you write a paper, someone read it and tell you how is it going, and you will do some changes and send another loop. The same will happen, but you will need to try to modify some biological signal. So when we talk about neurofeedback, really it's the same principle, but now instead of trying to modify anything in the body that could be the heart rate or the respiration, what we are trying to modify is the neural activity. So neurofeedback will have a self-regulation of the brain activity or brain state or behavior, right? And the goal is to alter the behavior or performance by means of somehow alter the mechanism, the neural mechanism that is behind that. So I will go over this schematic, and this will be the outline for what we are going to be discussing today. So we have a subject. We will need to acquire data, process the data, extract features, and then generate the feedback and put these within an experimental design. So the main point in the neurofeedback, or the critical point, is to define what is the feature that you're going to extract and display back to the subject. So what is that the subject is going to see or hear or feel, and how is going to alter him or her neural activation, system. So in general, what we have is a control condition, a series of neurofeedback trans, and then we will have a transfer where the subject will try to perform the same task that was done and control or during the feedback runs, but now without the extra information of the performance or behavior. In many cases, and we are going to get to that a little later, if we are talking about fMRI, and also could be done in EEG, we may, in most of the cases, people are collecting some resting state as well before and after these experiments to get a sense of where the subject is. So as I mentioned before, the first thing is the subject behavior or state, and that will be the brain activity. And by now, this is kind of familiar to everybody. The brain activity could be defined in different ways by the electrical signals. That will be the EEG or MEG, but metabolic or vascular responses, and in turn will combine into the fMRI signal. And this is the same that you have been seeing through the whole series of classes. And it's important to remember that, first of all, that the bold signal has a good spatial resolution but poor temporal resolution, while the EEG has the opposite, right? It's very good in time, however, the spatial resolution is poor. And however, if you think about where the signals are coming from, none of them, either EEG or fMRI, are a direct measure of the neural activity. There are integration of something happen in the brain. Still, it's a good proxy to understand brain activation and brain states. So the data acquisition in general is similar, whether we are doing neurofeedback with EEG or fMRI or both together. We need a standard EEG or fMRI system, with the adding requirement is that we need to be able to somehow extract the data on real time so we can move forward. In most of the systems, in our scanners, most of our scanners have this capability. Actually, all have them. In the GE scanners, we have integrated AFNI on the real time, and you see, if you're scanning on the GEs, you will see the time courses that pop up in the screen, and that data can be taken and put in the rest of the special programs that would allow you to then display it back, process and display it back to the subject. In the Siemens scanners here, we don't have it, but for example, Michelle Hampson in ATL, she takes the features of the reconstruction, the online reconstruction from the Siemens scanners and modify that script to get the data. So it's feasible in any of the scanners. So what we want to do, neurofeedback with EEG and fMRI, or together, they have different properties and different characteristics. So even though we are always trying to see how the brain works and how we can manipulate these signals. But EEG, the equipment, is relatively low cost and low maintenance compared to the fMRI. It's portable, can be put in any environment. So as a principle, we would like to be able to do, as a practical thing, to be able to do the neurofeedback with EEG. The signal is, while it seems easier, actually extracting the features or for the subject to manipulate those features is a lot more complicated. The training, we'll see some examples, but really if you try to extract what's unique when the subject is doing something on the EEG, the signal to noise is very low and you require a lot of averaging and the training is not that easy. However, the fMRI, even though it's very expensive, right, for a treatment, like you don't want to need to put continuously a subject there, but it allows you an easier way to extract features. The signal is smoother, and you can detect in an easier way, signals in single events. In fMRI, in EEG, I mean, there are algorithms and ways to detect single events features, but online will require a lot more training than what is needed in the fMRI. So anyways, the system for EEG neurofeedback would be, let's see if I can get the mouse. We have the stimulus computer, a subject with a cap with electrodes or whatever electrodes you want to put that will connect to an EEG amplifier that will be connected to an acquisition PC, and then you will have either in the same computer, that could be in the stimulus computer, in the acquisition computer, or a separate unit where you will do some pre-processing and feature extractions. So that always needs to be done. If we do the fMRI neurofeedback, again, we'll have, here is a figure pretty standard from Bob Cox in '95. So that's kind of the principle of having real-time fMRI. What we're doing now is extracting the data, as I mentioned before, that we are seeing in the screen, and we can decide which areas we are extracting, pre-process that, extract the feature, and use that to display to the subject. I guess the simplest neurofeedback will be that you just display this time course to the subject, right? And you, without doing anything more than plugging that in your computer, you will have a basic neurofeedback system in the fMRI. And if you've been in the scanner, like if you ask someone to do a motor task, you will see, if you put your cursor in the proper way, you will see that activation. So that will be kind of the basic, and an easier way to understand why it's so easy to do that. If you do the same with EEG and you ask someone to do a finger tapping, it's not that transparent what is the activation, even though you can process and you can know that the subject is moving, but you will have more detectable things in the transition from activation to rest and things like that. So now when we want to do both together, and the reasons could be different, I will talk a little bit later, we need to now have the same fMRI setup, the EEG setup, similar to what you saw in the lecture that Jen gave into how to have that in the scanner. And then your feature extraction will need to combine the EEG and fMRI data. And again, that will be not only extracting the feature, but also you will need to find a common space for these things. Remember, the time resolution and the space resolution of both are different. So you will need to figure out what features to combine and then project it back to the subject. So as I mentioned, the pre-processing will be, again, traditional, but in real time. So this is what I was just mentioning. If you were going to combine these two signals, you need to define what to look. And just as an example, when I mention real time, the question is, what is real time, right? How fast needs to be real time, right? Not a wait. And of course, if you're talking about fMRI, we are talking about longer delay because our signal is slower. And I like this scaling, right? If you have these ERP, event-related potentials, together with the fMRI, the time scale is completely different. So if we were sending back to the subject information about this peak in the EEG, you will get the signal within 170 milliseconds. However, if you're looking at the fMRI, the response to this will come a few seconds later. So those are things to consider when you are setting a neurofeedback experiment, and also how you will tell your subject that things will happen. So based on regardless of the time, when we do the pre-processing of the data for the EEG, again, is fairly similar to what you will do in a regular EEG experiment. You may want to do it in few channels at a time, depending on what strategy you're using, just to speed up the processing, right? The more data you try to process, the longer it will take. Of course, with EEG, that the data is collected pretty quick, and some of the operations are fast, so it won't affect much, but still you may want to reduce the amount of operations. So you will probably do some analysis in the time domain, especially in filtering on the trend in the data, cleaning it up a little bit from baseline corrections or ocular artifact corrections. You can do some transformations and decide whether the data is clean or not, like if the subject move and you have a signal that is like a fast muscle activity because the subject yawn or clench or move around, you may not want to include that, but anyways, you could decide on what type of pre-processing to do, and then you can finally do either trying to detect one of these peaks in the data, or you can do some frequency analysis if you do, and again, for this either method, you will be looking at a little window within the EEG, but you could talk about milliseconds, right? Hundreds of milliseconds, and you will be able to do this. You can also, if you were doing simultaneous EEG and fMRI, and your fMRI will be collected every second or every 2 seconds, you can use about the same time frame for that. You can look at, if you're looking at the frequency, you can look at the power at different bands. You can look at cross spectra. You can look at coherence, synchronizations and desynchronizations, and so forth. So as I mentioned before, the difficulty in these is that the signals are quite noisy. That signals that I showed you before are an average over a large number of events. People, I mean, in fMRI, if we have 20 events to do an average, it seems like we have a good number. In fMRI, in EEG, you go to much, much higher. Even though, again, once you have the average of the full run or the characteristics of that wave, you can try to use those features to try to detect what's going on, but it requires the training. So the signal to noise is low, and as I said before, it's hard to train on the subject. And this is a common issue with, you probably hear about brain-computer interface, where you're trying to get the subject to control a machine based on the electrical waves, right? And this is the same. I mean, it's not something easy, even though you can have all the electronics to move an arm or move a leg. It's hard to get the synchronization with the brain to just give that command. So this is the same principle. With fMRI, the limitations are slightly different, right? We extract the data from some brain areas. When we have the electrodes on the head, the subject move and the electrode move with the head. When the subject moves in the scanner, the voxel stays in the same place. The head is in a different one, right? So this is one of the problems that we have. The second problem is that when we measure EEG, we're measuring a voltage, right? That's an absolute measure. We know we can correct by impedances. There are a few things that you may need to adjust, but it's a metric with a unit. Ball fMRI is not, so we usually refer to a percentile signal change. We can refer an area to another, but we don't have a proper calibration for that. So when we try to do a neurofeedback, we need to figure out how we are going to deal with the variability that this signal will have. And as I showed in the previous picture, and we talk about, this hemodynamic response is slow. It would take seconds. So if the subject's, the brain activity is much faster. So if I'm not aware of what I'm doing and I'm trying to control my brain to increase a signal, and I don't see that happening, I may change my strategy before the system responds, right? Because it will take at least these 2 seconds. So it's something to be aware when you give instructions, and that's kind of a very important point in any neurofeedback. What do you tell your subject to do? So to run the fMRI study, we need to do a few things before really running the neurofeedback study. We need to have a region of interest definition. We could send the whole brain, but we need to define what is that we are sending and what is that we are looking. So in general, we have a functional, a structural data set and a functional run that could be either a localizer, just because we are using regions that were defined anatomically in the structural and then converted to the template, or we may have an actual task where we define regions of interest that then we were going to ask the subject to manipulate. So regardless of which one we choose, we need to select one of the EPIs. So if when you run your fMRI processing, you usually register everything to one particular volume within the series. When we do the online motion correction in the scanner, we need to, and this is very important, because if you're looking at a region of interest and your subject move, you are looking at somewhere else. So you need to make sure that the region that you are trying to use for the feedback is the region is continuously the same, because if not, the subject will control the feedback by just moving the head, right? So we need to make sure that the region moves with the subject, so as much as possible, and the subject move at the least possible. So we set up in the scanners the reference for the motion correction, the volume where we define the region, and that's usually in this functional data set that we register to the structural, whatever we define this RI. And then regardless of the run, we always refer back to that. So that that has the trade-off, because you want to train your subject for as long as possible, but the longer that you're in, the more likely the subject have moved, and the registrations may have some issues as well. And while a lot of these things you cannot verify everything online, you can always check how much each of the things would affect your data as you go by. And finally, as I mentioned, that the bold is a relative measure, right? So we need to somehow reference that signal to something, and usually we use percentage signal change, or we can do some reference with different ROIs. So as I mentioned, the motion correction, we fix by registering the data to the space where the ROI was defined. One of the problems is not only the non-absolute value, but also we have a physiological noise. And one of the ways to correct that is assuming that the amount of physiological noise would be similar, and you can use a reference region. Some people use the white matter. So there are different strategies, but it's always good to have some sort of correction or some other reference that will remove these things or would just diminish their impact. And then once you have that, then you can extract the values from the voxel and do different things, right? Just compute something on those voxels to extract a feature. So what feature we are going to extract, that will depend on the question that you have as a researcher. So the features in EEG, as I mentioned, could be one of the peaks in the ERP, could be the power spectra at a particular band, how the power spectra between one band changes, like how much synchronized or desynchronized get, and that, for example, is relevant to some motion. It could look at the coherence between different electrodes, or the ratio between power bands in different electrodes and things like that. And in the fMRI, we originally start all the groups looking at neurofeedback, start looking at activations in a particular area, activations in one area with respect to another. We are going to discuss a little bit in connectivity, why we get to try to modify connectivity nowadays. And I'm not going to talk a lot about this, but this is a work in progress here and other places where now not only we can look at activations or connectivity, but we can really detect a pattern and try to make the subject fit this pattern in a better way. So we can do a neurofeedback based on pattern analysis, multivoxel pattern analysis. And that's implemented also in one of the scanners here. So from the EEG, the literature goes back to probably 95, and I'm sure we can find some older things. As technology gets more advanced and computer power is higher, we start getting a lot more of that. This is just a recent example of a group just for no particular reasons, but just that illustrate this example. So this is a group interested in compare the frontal alpha asymmetry and see how that affect the negative effect and anxiety. So for this study, they base that the frontal alpha symmetry is related with withdrawal of motivation, is associated with the affective of each individual star. So they run neurofeedback training, collecting the EEG from the frontal electrodes in the right and the left, and evaluate whether the changes in the right and the left, and also how they respond to affective tasks, right? And this is done with 32 subjects, all females and right-handed, and divided in two groups. One group that they do neurofeedback, and one group that does not do neurofeedback, just do something else. And they show that there is an increase in asymmetry post-training in the people that went through the active process of neurofeedback and trying to control that, right? This is in the two frontal electrodes, right and left. They can, I don't know how, these two lines not fully separate with the errors, but definitely you can see that there is a disparity into what happened in one group and another. And when they compare what happened at the behavioral level, they also find significant difference in the positive affected scores. So that kind of suggests that if you manipulate the proper way, you can produce some outcome. How long it would last, that will be a different question. And so this type of experiments allow two things. One is to think of in terms of treatment. How can you improve the outcome of a patient? This, again, I mentioned, has been proposed for 20 years. I don't think is a strong evidence that the outcome is long-term. However, it allows to understand what things are affecting the mechanisms, right? Whether manipulating something really change the behavior, even if it's at that particular time, it could tell you that there may be a relationship, and you can start thinking of mechanisms or other ways to alter that pattern. In fMRI, I'm going to talk a little bit more about some of the experiments because they're newer, because it's an MRI course, and also because we have been doing some studies over the years. And there have been applications in different target areas at level of activation. Primary motor area is one of the first type of experiments, and as I mentioned before, if you put the cursor in the area, you could see the activation. So it's a very intuitive and easy target. It's easy to explain the subjects, and also we know that the activation in the motor cortex, in the area, in the motor system, I would say, not in the motor cortex. We'll get to that in a second. In the motor system, would increase if you are even thinking about doing more activity, right? It will increase if you do more activity, and also will increase if you think about it. So, but we'll get into details of that in a minute. Has been done in sensory areas, supplementary motor areas, insular motion network, and also there have been work in connectivity, both as a post-hoc analysis into what happened with the data while you're doing these tasks, but also some work that we have done, and others are trying as well, on making the subject manipulate the connectivity, and there is a strong reason for doing that. So a typical experiment, one of the first experiments that we did would be to have subjects first learn how to use how the system works, right? The delays and things, by first doing the motor task and then using the imaging of the task. So we can say, try to move a bar, thinking about movement, try to decrease the bar or suppress that by doing something else that wouldn't involve movement. And as I mentioned, like the motor cortex is just pretty easy to manipulate, and we can control. For example, if we're asking someone to do a finger tapping, we can put an EMG on the finger, and we can see that when the activity is going and when it stops, and we can see a very good correlation with brain activity. This is kind of pretty easy strategy. As I mentioned before, we also ask the subject to do the same with imagining, so this won't be happening, this will be flat. And we see that the subjects can modulate the network, but the motor cortex per se would not be activated during the imagining, which makes sense because you could activate the whole network, but the output should not activate, right? And these things are could have relevance in trying to work with people that need to go through rehabilitation, for example, because in those cases, you can't tell them to imagine moving while they're trying to move, and then you can see whether this motor cortex start having some activation. So, but the question that we have was whether, the areas decrease during the feedback. The motor cortex per se did not change during the imagining, but what happened with the connectivity? So we can, and this is done post hoc, right, with the same data that we can look at the motor task, and we can define a region of interest, right, see which area activate during the finger tapping, and then we can do the basic time course correlation of that seed with the whole brain or whatever part of the brain we were collecting data from. And then we can compare the connectivity values the same way that we do with activations for the feedback and no feedback runs and before and after training. And we can see that, as I mentioned before, the movement and the imaginary activations, right, during the go have kind of similar, but the motor cortex is certainly activated during the move and not that much during the imagining. However, when we start putting the neurofeedback, we start getting more of a network of correlation. So even though this is not active, we do have a strong correlation as we increase on the feedback, and when we transfer, when the subject is doing that without the feedback, still get that, because when we kind of reinforce the activity within those areas. And what we see is that the connectivity, not only in the motor cortex, get correlated, this got bigger, sorry, with certain areas that are interesting. So the anterior cingulate is related to the area, to the monitoring of the task, right? And we see that the correlation increase during that. And we also see that the connectivity after training was higher with the basal ganglia areas that are part of the same network. So these could tell us somehow what things are involved in the process of the neurofeedback. So one of the things that come out of this, and there is a follow-up with a group in Switzerland, that they collected the data from a group, a lot of studies that did neurofeedback, and then look at what is the effect of neurofeedback per se, right? Because why the anterior insula would be involved here. So they look, the anterior cingulate, I'm sorry. In that study, the anterior insula is one of the areas that kind of pop up in a lot of the neurofeedback studies. So there are things to consider when you do these experiments, that is, what is particular to the task that you're doing and what is inherited of the being monitoring a task and being trying to control what you're doing. So talking about the insula, this is another of the experiments that we did. We tried to see whether subjects could control the anterior insula, and the reason for that is that we think in Tourette's patients this would be relevant because they have an urge to move, like a perception of their own body and sensations, is, and the insula has some level of increase of activity as these events are happening. So if we can somehow teach the subjects to decrease the activation in this area, we could reduce the number of tics, and it's one of the projects that we are still working. But to start off, the IRB suggests that we start with healthy volunteers, so this is what we did. To find out the area, we use a blink suppression because this was healthy volunteers. So if you keep your eyes open for a minute, once I let you blink, you will blink a lot more, right? But at the same time, there would be an increase in the activity in the insula. So I don't know if you can see it well here, but this is a ramp, right? And each of these blocks is one of the blocks where we ask the subject not to blink. So we can define functionally which area would be related to the urge or like the pressure to blink. And then we can do the experiment where now we ask the subject to try to increase or decrease this area, and we can see how it goes and how well they can manipulate this activity. Now, not necessarily doing the blink suppression, that's just to define the area. And what we see is that some subjects can modulate to a certain level the activity. Some subjects are not that good, but these are the best subjects, right? You see here, the best performers, right? They can do it, but not everybody can regulate that. And I mean, it can be done, but it's like few out of probably half the subjects or a small amount, and it's hard to transfer. So, however, when we look at, this is like on the modulation of the activity is as the activity in the region that we predefine, and we are doing the feedback on the subject. This is anatomical, so all this is done post-hoc. However, if we look at the connectivity also post-hoc from this area, we see that really the subjects during the neurofeedback do increase the connectivity with the middle frontal areas, right? And that's something that we know that these areas are highly connected. So this middle frontal gyro is connected to the insula, and so it seems that there are other mechanisms that one can exploit to try to manipulate how the system is working, because also we know that in these patients, not only the activation is different, but we also know the connectivities are different. So in the same line, Michelle Hampson in ITL have also been working, trying to figure out how the connectivity work in areas that are relevant to Tourette syndrome. She tried to make the subject increase or decrease another of the areas of interest, that is the SMA. And again, in some cases, it worked, not always. The transfer is hard, but again, when she looks at the connectivity, again, she can observe the same way that we observe in the other cases, that there are changes in connectivity that happen after the training with the neurofeedback. And that's the reason why I was mentioning that it is, in most of the studies now when we do neurofeedback, one of the things that pretty much everybody's collecting some resting state before and after, because it will let us know not only the transfer, but really are we modifying the networks in the brain beyond what the task per se would be doing. And in these lines, this study is done by Lei Liu. She did some on healthy volunteers here with us. She was a postdoc in Leo Cohen's group, but this study was on stroke patients in Germany. And when we are working with patients, this works much better at the individual level, because there is, first of all, there is space to improve, right? When the system is working at an optimal level, trying to modify it is harder than when you have a deficit and then you have a possibility to change something. And in this case, she was looking at the connectivity between brain areas. And so you can see here that the control increases, the connectivity improves, and that correlates in the 4 patients that you see with the scores of the behavior. So other way that we can use the the fMRI and neurofeedback, we can use the EEG to understand the signal. So we know, and it's very easy to localize and extract a value, but we don't know the neural activation. So one of the reasons to try to put together an experiment that is so involving as to have EEG and fMRI and neurofeedback, it's to try to understand how all these signals relate to each other, because once you have that understanding, if you can identify what is changing in the EEG, maybe you can transfer the experiment to be run only on EEG. That would be much cheaper and practical to do on patient population. So this is a study done by Soteb working with Jerzy Badurka, and they look at the neurofeedback for the amygdala left and right, and then correlate that activity with the asymmetry changes in the EEG in a set of electrodes, right? And they can check whether the EEG in some frontal areas correlate with the fMRI in the amygdala. And of course, that's complicated, and you need to figure out which is the parameter that will correlate. But once you find it, that allows you to move forward in the experiment outside the scanner. On the same lines, we are in one of the protocols that we are working. Also, we're trying to figure out what is a good state in the brain, for example, for learning. So you can find out what is the patterns that correlate patterns of brain activity in both fMRI with behavior, and then figure out what EEG pattern correlates with either, and see whether modifying one or the other will produce the improvement in the outcome. Another way that the EEG and fMRI with, this is kind of aside, but kind of a refitting of the data is how you can use these signals not only to know where the activity is coming from, but also to correct for noise. This is on a side, right? You can get some extra information from the fMRI and EEG and improve your data. And if you can improve your fMRI data, you may be able to do a better neurofeedback as well, right? So all these signals are informing each other. So just to wrap up, there are a couple of recent review papers in terms of what has been going on in the neurofeedback literature, and from this paper I extracted some of the prerequisites for a good neurofeedback study. Since it's already written, no need to rewrite it. And so we talked before that one of the critical things is to get a feature, right? Define what is the feature that is relevant for the study that you're doing, and that could be chosen based on other EEG or MRI studies, on the literature, on figure out what is relevant for the question of interest. Then you need to figure out whether you can train a subject on that feature, right? Can it be modified, right? What is the number of subjects that you will need? How many runs the subject need to perform to to be able to modify that is doable, right? If I need to have someone for 8 hours in the scanner 5 days a week, probably won't be practical. If the person can be in the scanner an hour a day for a few days, it may be doable at least until we train and find out how to do it outside the scanner. And one of the features that is important is that we can transfer the performance. As I mentioned before, you will have a control, so you know where the subject was, do all your neurofeedback runs, and at the end, you would test again the subject in the initial condition and see whether there was an improvement. And in general, that is compared as well, that ideally, with some behavioral, some performance. So not only that the brain activity changed, but did, I don't know, if it was a motor task and I'm doing a pegboard moving pieces from one place to another, if my connectivity increased, did my speed increase, right? Of course, when you get to healthy volunteers, one of the trainability of the feature would be if I'm at the top performance, there's no way that I will improve, right? So you need to figure out if a task that have room for improvement before you can do it. One of the important things is to have a control. So that's usually a sham or pseudo feedback. That is, you have someone doing exactly the same routine, but are not showing the proper data. So the neurofeedback won't be happening, right? That external cue won't be happening. And there are two ways, I mean, and you can argue in favor of using a sham signal that will give the wrong feedback, or performing the task without the feedback or with another type of feedback, because if the feedback really works, and you're showing something different, you may be taken out of, right, you're confusing the subject and you want to have more of a neutral thing. But that's a balancing depending on the task that you're doing. And of course, you hope for a random assignment of the subject, and ideally in your control conditions, both groups should be similar, right? If you have a significant difference at the baseline condition, then it's a problem, and of course, in many cases, you cannot predict that if people are randomly assigned. But it's important to check. One option is to divide up the subjects after doing an analysis of the control and randomly separate them to get similar values. But you do need to check that the control is similar for both groups. And from another paper that also, this is a little older, but there are some changes in the literature, but still some of the challenges, I mean, there are more progress, but the challenges keeps on being similar. So can we use these techniques to treat neurological disorders or psychiatric disorders? There are, as I mentioned, there's on the both fMRI and EEG more than, I mean, the EEG will probably 20 years, and a lot of the reports will be single case, some would be a little bit more solid, but still not a standard treatment. With the fMRI, there are some also results in terms, for example, the cases of the stroke patients. Some other disorders show some possibilities of controlling an OCD and Tourette, so forth, but still there is no standard for treatment. So what will be the conditions and the ways around to make this a tool that will help improve the clinical outcome, right? When will be these more beneficial than other interventions? As I mentioned before, many times we try things first on healthy volunteer before going to the patients, but not always this approach would lead us to the right answer, because there is a deficit in the patients that we are trying to correct, and now we are testing things on a healthy volunteer population where the deficit is not there. So we may not be able to manipulate things in healthy volunteers that we would be able to manipulate in patients. Whether these can be translated, like the transfer to be able to do outside the scanner, outside the EEG, and in the regular life. How effective is the treatment, how long these will last. Side effects, maybe, and how many times we need to go through these to be able to get to an effect that will last over time. And so with this, I would end and take questions.
What is the strongest data that convinces you that the fMRI neurofeedback is better to learn for healthy volunteers than the
Well, I think it's easier to control, right? If you try to get a subject to learn to improve, if you try to detect what is changing in the EEG when the subject is imagining a movement, you will need to do a lot of training on machine learning type of things, like to figure out what is the pattern in this particular subject and how to detect that signal, and then identify it on a single run to be able to tell the subject what it is. If you try to do in the fMRI, it takes you no time, right? You put the subject there and you will see the signal, and the subject is doing it and you can see it. So it's a matter of practical for implementing, and technique itself, right? The signal to noise in the EEG is lower. So when we start doing neurofeedback, here we started in, I think, 2009. I know people in EEG have been trying for much longer, right? And there's always a lot of programming and a lot of struggles to try to get the signal to either for single event detection or brain-computer interface and so forth. And when we start trying on the fMRI, it just works, right? There's a different question on how much a subject will be learning and how long it will take for that to transfer. But in terms of setting out the experiment and see how the signal is changed is just much faster, because you're looking at a bigger area, like integrated over space, and more secluded in space and integrated over time, right? But if you try to see from, just to give you an example, right, there are some experiments. If you try to see whether someone is thinking about a scene or thinking about moving, if you put in the scanner, you will have a very clear pattern, right, on the motor system or the visual system. If you try to do that with EEG, you'll have to do a lot more work to separate the signals. Yeah.
For how long have the longest reported neurofeedback results or outcomes lasted?
I don't think there is very long, right? People are still, I don't know whether people are doing follow-ups in the rehab patients. Probably if the patients gain some function, it may stay, but I don't know the times. It's scarce.
And regarding the referencing, because most neurofeedback studies are over several days, do you then choose the reference for getting the percentage of activation of the whole signal every day again? Or do you take the baseline of the first day?
So you're using, usually you don't necessarily use the baseline value, right? Because each time you put the subject in the scanner, that's why, I mean, if you're talking about bold signal, the actual number that you're measuring, it could be completely different, right? It's not like you put the same subject in the same place, you're getting the same value. So you can get a percentage signal change of whatever is your baseline, and you still will need to collect some localizer to register. What I would say is that, yeah, you need to use the same region of interest, unless you're doing a functional localizer every single time and see how the functional localizer change depending on your question. But I think I would use the region the same if I'm trying to study, but the value will come from the values of that session. And usually will be either percentage signal change, the ratio between areas, or connectivity between areas, right? Or a pattern. If you use a pattern, probably you do want to use the same pattern if you think that pattern is correct. And actually, in one of the experiments, we are trying to work on a pattern created from an fMRI study on the population, like on a group of whatever 20 subjects, and then transfer that back to each subject and see whether they can reproduce that. So it depends on your question. Okay. No more questions, thank you.